Welcome. In the last session, I discussed about R.H. Whittaker's five kingdom classification, and now in this session, I'm going to give you characters of each and every kingdom. Now, before we discuss those things, here I would like to tell you a few more points. In 1977, Carl PUC et al. He proposed three domain system. He proposed a three domain system, and those three domains are domain eukarya, then domain archaea, and then domain bacteria. So these are three domains. So the kingdom Monera, so they divided into two groups, and these two groups will include prokaryotes. So these two groups include prokaryotes, whereas this domain eukarya, it includes the remaining four kingdoms. So all those remaining four kingdoms will come under domain eukarya, whereas the archaea and bacteria will be the prokaryotes. So this domain will come above the kingdom, uh, taxon. So we know that the taxonomic hierarchy, it all starts with the species. Then above that we have genus, then family, then order, class, phylum, kingdom. So we know there are seven obligate categories. So these are seven obligate categories: 1, 2, 3, 4, 5, 6, and 7. Now above this kingdom we include the domain, so that becomes the eighth category. So this is domain. So domain will consider above the kingdom. Okay, so this is recent classifications. Now with this, it has become a six kingdom classification. So six kingdom classification. I told you already in my previous videos that the classification is not permanent and it will change from time to time. So here, coming to the six kingdom classifications, we have is kingdom Animalia, then kingdom Plantae, then kingdom Fungi, then kingdom Protista, and then kingdom Archaea bacteria, and then lastly kingdom Eubacteria is Eubacteria. So you have, okay, so 1, 2, 3, 4, 5, 6. So this six kingdom classification. So this is six kingdom classification, right? And last, okay, Archaea and Eubacteria, these two are prokaryotes. The remaining four are eukaryotes. So that's what I have mentioned here. So this eukarya, so this belongs to domain eukarya. It includes the four kingdoms: Animalia, Plantae, Fungi, and Protista. And okay, the domain Archaea, it includes Archaebacteria. So this comes under, right, domain Archaea. Okay, whereas this one comes under domain Eubacteria. So that one comes under domain Eubacteria. Okay, I'll mention here. eubacteria. Okay, so of course I'll just the meaning of archaea, so from Greek archaea, which means ancient, and we already know here from the Greek eu means true. Okay, and so here the first organisms to be present on the earth are eubacteria, and that in course of time have given, okay, or diversified to form archaeobacteria, then the protista, and then the protista got diversified to form the fungi, plantae and animalia. Okay, so these eubacterians, they got diversified and have given rise to different groups. So this is the six kingdom classification. Okay, so now that's about a small information I would like to give you. Now we will see each kingdom in detail, one by one. Okay, I'm not going to discuss this six kingdom classification. I'm going to follow the same five kingdom classification. So I'm going to first discuss about kingdom Monera, and then kingdom, okay, Protista, and I will go in this way, not with the six kingdom classification. So this is a small information, okay, about the six kingdom classification. And I told you we even have seven kingdom classification, and these classifications are not permanent and they will change from time to time. Okay, so now let's see about kingdom Monera. Right, so I already mentioned that the kingdom Monera, it includes all unicellular prokaryotic organisms, includes unicellular prokaryotic organisms. And I already mentioned in the last session that the prokaryotic, which means these are the organisms who don't have nuclear membrane. So prokaryotic organisms do not have nuclear membrane. So if they are not having nuclear membrane, so that is no nucleus. So they don't have nucleus, and their body is made up of only one cell. So that's we call unicellular. And all those organisms that are included under this kingdom are so small that we cannot see them with our naked eyes. So they are all microorganisms. So all these are microorganisms, right? And next, so these organisms belonging to kingdom Monera, so these are cosmopolitan in distribution. Means, so here cosmopolitan in distribution means they are present everywhere. So cosmopolitan in distribution, which means they are present everywhere. So they are present everywhere. Okay, so they are present in the ice in the North Pole. Okay, and in deserts they are present, in hydrothermal vents they are present, whatnot. There is no place that we cannot say these microorganisms are absent. So that's what we say, these are cosmopolitan in distribution. So that is, I'll say, they are present in soil, they are present even in water, and they are also present in extreme habitats. So they are present in extreme habitats. Habitat, which means, okay, location where the organism lives, where the organism lives. So they are also present in extreme habitats. So those extreme habitats like hot springs, so where the temperatures will be very high, more than 100 degrees Celsius. Even in those places also you will see the presence of these bacteria growing. Okay, and then we will also see in deserts. So there also the temperatures will be high, dry conditions will be seen. Even in those locations also you will see the presence of these group of organisms, that is monerans. Then okay, in snow also we will see, and even in deep oceans, in deep oceans also we will see the presence of these organisms. So these are all extreme habitats where we will find the presence of these unicellular prokaryotic organisms, which we commonly call them bacteria. Okay, so next coming to mode of nutrition in monerans. We find two types of nutritions, one autotrophic and also heterotrophic mode of nutrition are seen in this group of organisms. Under autotrophic, we know we have two types, photoautotropic and chemoautotropic. Right, so here photoautotropic, which means, so here you can see from the Greek photo, which means light, then autos, which means self, and trophos, which means nourishment. Right, so this is self nourishment but using the source of energy, the light. So here the source of energy for preparing the food materials is light. Coming to chemoautotropic, here the source of energy is inorganic compounds. So one example for these inorganic compounds I would like to give, the inorganic compounds like H2S, so that is a hydrogen sulfide. So now how using that hydrogen sulfide these organisms are able to synthesize food. So I'll give you a equation, right? So H2, that is 18 H2S plus 6 CO2 and 3 O2 will result in the formation of C6H12O6. So this is the carbohydrate. This is the food that they have synthesized using hydrogen sulfide. Okay, and in this process we get some water and right 18, yes, that is sulfur is released in this process. So I just gave this equation for sake of understanding only. Okay, so this is how chemoautotropic organisms synthesize their own food materials. So chemoautotropic and photoautotropic both are under autotropic mode of nutrition. Then coming to heterotropic mode of nutrition, so we see here there are two modes, one saprophytic and another one parasitic. Now we will see both saprophytic, what actually the saprophytic mode of nutrition, and also parasitic. Now in case of saprophytic organisms, so what they do, so when they grow on a solid food substance, so this is a solid food substance, right? So when these organisms, the bacteria, grow on them, so these bacteria are going to release enzymes. So these bacteria growing, and they release enzymes onto this solid food substance, and because of this, the solid food substance will get converted into liquid food substance, so you can see the liquid food substance, right? Now this liquid food substance is absorbed through their body surface. So they absorb this liquid through their body surface. So this mode of nutrition is what we call saprophytic. And coming to parasitic, in parasitic we have a host that harbors the parasite, and parasite is a organism that takes shelter in the host. So it depends on the host for food, shelter, and also for the purpose of reproduction. So for reproducing also it depends on the host. Okay, so host harbors the parasite. Okay, in coming sessions we'll understand in detail the parasitic. So whatever that nutrients the parasite requires, it takes it from the host. Okay, so these are two modes of nutrition that we see in case of Monera, autotrophic and heterotrophic. Then coming to reproduction, these organisms reproduce both by sexual and asexual mode of reproductions. So here the reproduction in these organisms is by asexual methods and also by sexual methods. The asexual methods include, like for example, fission. So fission is a process where a bacterium, okay, so is a process where a bacterium divides into two daughter individuals, so by means of fission, right? So these are daughter individuals and this is parent. Okay, and coming to sexual methods, it includes like transduction, then transformation, and then conjugation. So these are three modes of sexual method of reproduction in case of bacteria: transduction, transformation, and conjugation. So let's see what actually is this transduction. So coming to this transduction, so it means the transfer of that genetic material from one bacterium to another bacterium by or through a virus. So this will be transfer of genetic material. So this genetic material is DNA. We know the full form of DNA is deoxyribonucleic acid. In bacteria, the genetic material is DNA. So transfer of genetic material from one bacterium to another bacterium through virus. So it means that the virus will carry the DNA from one bacterium to another bacterium. So if it is the case, then such a mode of sexual reproduction is called transduction. And coming to transformation, in case of transformation, so here just I'm telling you briefly about the meanings of these words. In our coming sessions, we will deeply understand about these words and even genetic material and even DNA. Okay, so step by step. So here I'm just giving you a brief idea. Okay, so transformation is uptake of the genetic material by the bacterium from its surrounding environment. So this is uptake of genetic material. I already told you genetic material is DNA of genetic material, okay, by the bacterium from its surrounding environment, from its surrounding environment. Okay, so say here this is a bacterium and this is a DNA, and this DNA will get into the bacterium and becomes the part of its genome. Then such mode of sexual reproduction is called transformation. And conjugation is the transfer of genetic material between two bacteria. So here two bacteria come side by side and they form a conjugation tube, and then they transfer the genetic material from one bacterium to another bacterium. If it is the case, then we call it conjugation, right? So conjugation is transfer of genetic material from one bacterium to another bacterium. So this is transfer of genetic material from one bacterium to another bacterium. Okay, so this is one bacterium, this is another bacterium, and both will form a conjugation tube that will help in transfer of the genetic material. So this genetic material is DNA. So you can see here the genetic material is being transferred to another bacterium, and such process is what we call conjugation, right? So that's about the different modes of reproduction in bacteria. Now these bacteria are classified based on their shapes. So types of bacteria based on their shape. So we find here four types of bacteria based on the shape. One, okay, bacilli, then coccus, right, or I'll mention cocci, and then spirali, and then vibrio. So these are four types of bacteria. Actually I used plural words. Coming to singular, okay, so they will be, so they are called, right, bacillus, right, so singular bacillus. Then here also in case of cocci it is coccus, and spirali is spirillum, and then The Vibrio is Vibrium. Now, what is the shape of these bacteria? So let's see, the bacilli are the rod-shaped bacteria. So they are rod-shaped bacteria, and they are seen in this way. So all these are rod-shaped bacteria. Okay, then coming to cocci. So these are spherical shaped bacteria. So these spherical shaped bacteria are seen round. So these are spherical shaped bacteria. And then spirali. So these are spiral shaped bacteria. So you can see, so this is how the spiral shaped bacteria are seen. So those are spiral shaped bacteria. And coming to Vibrio, so these Vibrio are the comma shaped bacteria, and they appear in this way. Okay, so they are comma shaped bacteria. So these are four types of bacteria based on their shape: bacilli, cocci, spirali, vibrio. So here on this side I have mentioned all with the plural words, and in the bracket I have mentioned singular. So bacillus, coccus, spirillum, vibrium, and this is the shape of those bacteria. So four types of bacteria based on their shapes. Right, so with this, I will end today's session. In the next session, I'll discuss about archaeobacteria and eubacteria in detail.